All right, so now we are in chapter three of sorry, not chapter three, part three of chapter 26. So in this part, we are going to go over what themes occur in diversifications of bacteria and archaea. Okay, so we're going to look at the genetic variation through gene transfer. We're going to look at morphological diversity, metabolic diversity, and ecological diversity as well as global impacts. Okay. So for the types of genetic variation through gene transfer, we're going to go through all of these. And I have some diagrams to show you that will probably that are more efficient at showing you the types of gene transfer. But the first one is lateral gene transfer. So this allows for the acquisition of traits that would not be available through binary fission. So binary fission is a type of asexual reproduction. That's where the cell of a bacteria would split into two. Okay? The next one is transformation. So in transformation, you have donor DNA that's released by a dead cell, so a cell that lysed, and it releases DNA into the environment. So then that DNA that's released finds its way into a new host. And once it gets in, the new DNA is incorporated into the living cell by recombination. So so the cell is then going to have, it's going to be transformed and it's going to have the recombinant DNA. Okay, so this is DNA transfer by transformation. The next one is DNA transfer by transduction. So this one involves viruses. So we have a bacterial phage, which is the virus that infects the host. Okay. Then it produces phages with both viral and bacterial DNA, and then it infects the new host. Okay, and then we have transduced cell. We have a transduced cell with recombinant DNA. So one more time. So the bacterial phage is a type of virus that infects either bacteria or archaea, and it also uses them for reproduction. The other one was conjugation. So there are two types of conjugation. So you can have two bacterial cells that come into contact with one another, and one cell contains a plasmid, and then with this structure called the conjugation tube, it sends a copy of the plasmid to the other cell, and now the recipient cell has the plasmid. The other type of conjugation, so you have the two cells. You're also going to use, they also use this conjugation tube, but here you just have a portion of the chromosome that makes its way through the conjugation tube into the other cell. And then the transferred portion of the chromosome recombines with the chromosome and the recipient cell. And now you have a recipient cell that contains the recombination bacterial chromosome. Sorry, the recombinant bacterial chromosome. Okay, so it has a little bit of both now on the chromosome. Okay, so now for morphological diversity. So bacteria and archaea are very diverse. So they're diverse in size and they're diverse in shape and arrangement. Okay, they can come in shapes like filaments, spheres, rods. They also differ in their motility. Some have flagella and others move by gliding. Okay, and here's just some examples of the diversity uh, for the morphological diversity. So you can see here they give you an example of the size comparison. So the smallest one, Mycoplasma mycoides, this one's about 1 micrometer in diameter. But then you can have like the largest bacteria, Theomargarita uh, namibiensis, which is about 100 micrometers. That's between 100 to 300 micrometers in diameter. So a huge range. And then here are the different shapes that they could come in. And then here's the motility. So here you can see the flagella. The other thing, the other thing that they differ in is the composition of the cell wall. Um, and the way that you can tell the differences in the cell wall is by the way that the cell wall reacts to a stain, which is called the Gram stain. So the Gram stain is used to examine the cell walls because when you use it, they uh, turn a different color. So when it's a gram positive bacteria, that means that the cells look purple under the microscope. So the cell wall, that means that the cell wall has a lot of peptidoglycan. Okay, so it has a molecule called peptidoglycan. When it's a gram negative, the cells are going to look pink, and that's because the cell walls of the bacteria have a thin layer of peptidoglycan, and they also have an outer layer of a phospholipid bilayer. So here you're going to see examples of gram-positive bacteria in dark purple, and the gram-negative in pinkish, in a pinkish color. Okay. So A, this is the gram-positive, so it has a thick layer within the cell wall or making up the cell wall. The gram-positive cell wall is made up of peptidoglycan. Okay, so a thick layer of peptidoglycan. Whereas in the gram-negative cell wall, there's only a thin layer of peptidoglycan. Okay, and there's also the outer phospholipid bilayer. So this is why you can't really see a lot of the stain, so it looks pinkish. Here you can see a lot of it and makes it look purple. All right, so moving on to metabolic diversity. So all organisms must acquire chemical energy that's used to make ATP. And they also have to obtain carbon compounds that they use as building blocks to synthesize cellular components like DNA, RNA, um, you know, proteins, and things like that. The bacteria and archaea use one of three sources for energy to produce ATP. So they can use light, and in that case, they're called phototrophs. They can use organic molecules, which makes them a chemoorganotroph, or they can use inorganic molecules that makes them a chemolithotroph. Okay. So here's the different types as far as ATP production. Okay. Because remember, there's two parts. So there's the ATP production and also how they acquire carbon. Okay. So for ATP production, like I said, if it's a phototroph, that means that they use light If it's a chemoorganotrove, that means they're oxidizing organic molecules. So these are molecules. If you ever see a molecule and it has a carbon in it, like um, it could be like methane, uh, carbon dioxide. So if the molecule has a C for carbon, it's an organic molecule. If they oxidize inorganic molecules, they're called chemolithotrophs. So now, so autotrophs, they synthesize building block compounds from simple starting materials. So they get their carbon from the environment, right? For it could be from CO2, it could be from methane. Heterotrophs, they absorb building block compounds from other organisms. Okay? So they get their carbon from organic sources. On this table, you can see all the different combinations for obtaining carbon and energy. Okay? So when you're determining whether the type of method, you always want to start by first identifying the source of ATP, okay? Because that makes up the first part of the name, and it makes it simpler to describe. Okay, so if you're given the sources, like here, these are all examples. Okay, but they give you the sources. So the first thing you would want to do is identify the source of ATP, because if the source is sunlight, then you know that the first part of the name is going to be the phototroph. Okay, so it's going to be a phototroph. If the if they're getting ATP from chemicals, then you know it's going to be a chemotroph. But if it's from chemicals, you have to determine whether they're organic or inorganic. So if they're organic, you call it a chemoorganotroph. If they're inorganic, you call it a chemolithotroph. Okay, so that would be for the first part of the name. For the second part, you have to determine whether it's self-synthesizing CO2, methane, or other simple molecules to create those carbon-carbon bonds, or is it getting the carbon from other sources, from organic sources? Okay, so I'll walk through a couple of these with you guys just to see if we can uh, make this easier to understand. So for example, so obviously the labels are there, but I'm going to just walk walk through the process with you guys, and hopefully that'll uh, make things more clear for you. So for example, if I got, uh, let's say, yeah, we could start at the bottom. So let's say this was the example that was given. So an ocean-dwelling Nitroso humilis maritimus, it's a type of archaea, is using ammonia waste excreted by fish as an electron donor and CO2 as a source of carbon. So if it's using CO2 as a source of carbon, okay, well, sorry, let's start at the beginning. So if it's using, so first we want to determine what is the source of ATP. So here they're saying that the source of ATP is ammonia, okay? So if the source of ATP is ammonia, that's a chemical. So we know that it's going to be a chemoorganotroph, okay? Now we have to determine whether, sorry, let me start over. So we know that it's ammonia. We know that it can't be phototroph, so because it's not getting the ATP from light. So it has to be a chemotroph. But now we have to determine whether it's organic or inorganic. So ammonia does not have a carbon in it. So this is an inorganic molecule. Okay. So because this is inorganic, then it would be a chemolithotroph. As far as how it gets the ATP. Now for the second part, we have to determine whether it's getting the carbon uh, from the environment, like CO2 or methane, or is it getting the carbon from organic sources, so getting it from other organisms. What it says here, the CO2 is as a source of carbon, so it's getting it from the environment. So that means that this would be called a chemolitho autotroph. Okay, let's do another one. So if we go up here, so it says heliobacteria and waterlogged soil, both photosynthesizing and absorbing organic. Okay, well, it says photosynthesizing. So for the first part, how is it getting the ATP? It says photosynthesizing, so that means that it's a phototroph. And then the second part is how does it get what's the source of carbon? Is it getting carbon from the environment or is it getting carbon from organic sources? Well, it says absorbing organic. So that lets you know that it's getting it from other organisms. So this would be a photoheterotroph. And then I'll do one more. Oh, let's do E. coli. So it says E. coli bacteria using organic matter and animal intestines as a source of carbon and energy. So it's getting both from carbon. So question one is, where does it get the ATP? Oh, sorry, where does it get the energy for ATP? Well, it says carbon. So we know that it's a chemotroph, and because it's carbon, it's a chemoorganotroph. And now the source of carbon for the synthesis of complex compounds. So this question, well, is it coming from the environment, like CO2 or methane, or is it coming from other organic sources, from other organisms? Well, it says here that it's coming from the organic matter in animal intestines, so that's from other organisms. So this would be a chemoorganoheterotroph. Okay. So I would go through these and either block or cover the labels and try to walk through these and see if you can come up with the right labels. Okay. So remember, you always start by first identifying the source of ATP. Then once you know that, then you move on to the source of the carbon-carbon bonds. Okay. So whether it's self-synthesizing from CO2 or and methane, or is it getting the molecules from other organisms? Okay. Okay, so we'll pause here and then we'll continue next time.